And usually we have about 10 people or so. It really kind of varies depending on people's schedules. And again, most of your audience is going to be the members of ARCS Illinois chapter, which are women who sometimes are scientists and sometimes are not. So it's, again, this is a good practice for a kind of a general public sort of a lecture.
Yeah.
I can see you in like slideshow mode. So like I can see all your little
Yeah, and if
so you're going to go into, actually, yeah, that's what I meant. Yeah, that's what we want to do, is the big slide there. Okay, fantastic. Let's see. Alrighty, I'm going to mute myself, and I'll come back in a couple of minutes after I finish up some other emails.
Mm-hmm.
Okay. Now don't panic. I'm gonna. No, huh? Hello.
Hi, Donna, how are you?
Oh, there we go.
Yeah, I can't see you, Donna. I can see your name on the screen.
Oh, am I? Oh.
So your camera's not on, but
Now there I am.
Oh, there.
Oh, look at this vision of loveliness.
You are a vision of loveliness always.
Hello, Anthony. I'm Donna from Chicago.
Hello.
So
I'm
excited to hear from you today.
excited to get to speak. Yeah.
One of the best, absolute, by far and away, about the best thing about ARCS is that you get to hear from and meet scholars and the work that you all do. And I'm not a science person, so I will probably understand less than almost anybody in your audience, but I love hearing what you have to say, and it's just such incredible work that's being done. So it's really exciting. So it's nice to be able to hear what you're going to tell us, especially osteoarthritis for those of us who are aging. This is kind of a nifty topic.
Yeah.
Sorry,
Yeah,
Pat. Okay. stopped you from saying something.
Oh no, that's great. I think it's yeah. I was just going to say that we will wait a few more minutes because I think there were other people who registered, so we'll give them a few more minutes to join us.
Okay.
And then, of course, just is if we were there in person, we could all unmute our microphones and have a little chit chat. So that's totally fine. We can do that right now. Donna, as you mentioned, arthritis is something that many of us think about and may have to cope with in our daily lives. And
Yeah.
the exact question of how much is it due to genetics versus how much is it changeable by lifestyle or that kind of stuff is like a key question.
Yeah. Yeah. I'll tell you, I love getting older. Life has been lots of fun. It continues to be fun, but little things like arthritis are not a lot of fun, and hearing and sight and those kinds of changes. So, but anyway.
I remember reading a novel a long time ago where it took place in Chicago. It was by John Powers, the guy that wrote "To Black Pat and Other Shoes," really effect up.
Oh yes.
So he's like growing up Catholic in Chicago, and so that was a trilogy actually. So there was when he was a little kid, it was like The Last Catholic in America. Then the teenage years was the Black Patent Leather Shoes, and then the last one was called when he was an adult. A different character was an adult, and anyway, in the novel, the main character gets to know somebody who goes by the name of God. So he would write letters, and God would answer them. But one of the letters he wrote to God said, "God, why do people get old? I mean, it's so terrible to get old,
Yeah.
And God wrote back to say, I don't know what you're talking about. Old people are my human sunsets."
Oh, interesting.
So it was a whole different perspective on aging and how we are evolving to be something better, maybe.
Yeah, I love it. I love it. I remember those books a long time ago.
Yeah. Yeah, those were great. But then after those 3 books, he never wrote anything else, and he became a motivational speaker.
Yeah. Well.
So I see Sarah's coming online with well, hopefully soon. I don't see the video feed here.
I'm here.
Oh,
yeah, I have to
Sarah.
work, so I'm not going to join video today, but I'm listening. Hi, everyone.
Hello.
Hello. Okay. Let's see. I gotta check my other timekeeping device here. So what do you think, Sarah? Should we get started, or should we wait for a few more people? I'm not exactly sure how many you're coming.
Let's give it another minute. Is Kate joining today?
No, Kate. So I'm moderating because Kate is our usual moderator, and she had a work thing that she knew would not be done by 5. So she's hoping she might be able to swing by at the closer to 6, maybe.
Let's give it one more minute, and then we should start.
Okay. But yeah, here at the Beckman Institute at the so Anthony, I'm down here at U of I Urbana-Champaign, and I work at the Beckman Institute, as you saw from my email this morning. And we actually do a lot of research on what we call cognitive aging, which is focusing on aging humans and the changes to their cognitive functioning. So for instance, some kinds of knowledge grow with aging, like more facts about stuff. So there are some kind of psychological tests you can give to show that people improve these things. But then there's a lot of other data that shows various kinds of declines in executive function or processing speed or other sorts of cognitive measures of reaction time and accuracy in memory or strategies in decision making and things like this. But a lot of the interesting work actually takes the perspective that, you're a being in the world, so you want to not just study the organism like the older person, but you actually want to build an environment that helps them be effective. So
it plays to their strengths.
Right. So so you think about the human technology or the human environment system, and instead of just saying, "Oh, you get older, so things get bad," like how bad does it get? Instead, you try to create an environment where we have like a lifestyle engagement sort of a model. Although still, if you take a kind of a classic cognitive psychology perspective, you just see scores going down, and then again, part of the promise of various interventions is to boost those scores, and so that's why you see a lot of dumb commercials for brain training software and cognitive games on your phone and stuff. And actually, Liz Steinmoro, who leads this effort at Beckman, she wrote a really great editorial, I think 2 years ago, with a bunch of other psychology professors, saying that all these cognitive brain training advertisements are really kind of a scam, and there actually is no good evidence that it really helps you. I mean, if, for instance, you play Scrabble Go like I do, getting better at Scrabble Go means I'll be better at playing Scrabble, and that's kind of it. You know, it might increase my vocabulary by a few interesting words, but it's not going to be like, I'm not staving off the effects of aging by playing Scrabble a lot. But basically, though, being engaged in doing something is really good for you. So definitely, you know, not just being a passive recipient of content, but actually engaging, playing a game, you know, reading. She actually does a lot of studies on reading and how that's really important too. So yeah, there's a lot of good stuff out there in the world about how to age gracefully.
Yeah.
So perhaps we can have actually, we might think about all kinds of other events we could do with other speakers as well as our scholars. There's a wealth of people out around.
Yeah, that'd be great.
Okay, well, we've given it a minute now, so I think we can go ahead and get started. So welcome everyone, my name is Patty Jones. I am the treasurer of the ARCS Illinois chapter, and I'm also the Associate Director of Research at the Beckman Institute down here in Central Illinois at the University of Illinois Urbana-Champaign. And as you know, we are having these occasional Fun at Five talks that we're doing virtually, having a real glass of wine or beer with a mutual talk. And we're very pleased today to welcome Anthony Hung to give us our next seminar. So Anthony, let's see, where is my little, so Anthony did his bachelor's in biology with a minor in global health from Duke University in 2017. And he is currently pursuing a joint MD and PhD at the University of Chicago in the area of genetics, genomics, and systems biology. And as you can see from his title on the screen, he'll be giving us a talk tonight about the effect of genetics on osteoarthritis. So virtual clapping to welcome Anthony. Welcome, Anthony, and take it away.
Thank you for the kind introduction, Patty. Yeah, I also want to thank everyone who's here today. It's always a pleasure and an honor to be able to speak to other people about my work who aren't either in my lab or go to the same school that I do. So today's talk will be structured in a way that hopefully will give something for everyone. I wanted to start off with a brief introduction into the field of biology, the flavor of science that I do in my lab, and why it might be important and might lead to interesting ways to seeing our world, before kind of giving an overview of the work that I'm currently involved in, and closing off with some forward-looking thoughts about what next steps I intend to take with my research. So as was already mentioned, I'm going to be talking today about the relationship between genetics as well as environment into whether or not someone has a predisposition for developing osteoarthritis. So to give you an overview of what I do in my lab, fundamentally, we're interested in kind of studying the human body, of course. And the human body, like all other living things on the planet, is made up of these little components called cells. Here I'm showing one particular cell that may make up one part of your body. But of course, as you look to myself or look to yourself, you know that not all of our cells are exactly alike. We have skin cells, we have hair cells. Within our body, we have cells that make up each organ within the body. And each of these cells has their own specific function and their own specific kind of activities that it's engaged in. So a fundamental question within our lab is trying to understand what causes the difference that we observe between different cell types within our body. What makes a brain cell different from a heart cell? And what is the underlying genetics that kind of make that happen? And in order to kind of get at these types of questions, the field of genetics kind of takes advantage of what is called the central dogma in biology. You can think about the central dogma as the flow of information that controls how genetic information becomes proteins or makes its effect on an organism. So in order to understand the second dogma, I need to talk about 3 different molecules that exist within each of our independent cells. So if you take any cell within your body and then zoom in on a particular part within the center of the cell called the nucleus, you'll find a certain molecule called deoxyribonucleic acid, or also known as DNA. And you can think about DNA as kind of the recipe book that contains all the recipes that are required to build up an organism, such as yourself or me. So within this recipe book, there are basically what are called genes or recipes to build either structural proteins or components that make up our skin, such as collagen, or our hair or nails, as well as the instructions for making small molecular motors that kind of control many of the activities that we engage in our daily lives. The second molecule that is found within each of our cells is RNA, which is very similar to DNA, but instead of being double-stranded is a single strand. And carrying forward this analogy of DNA being the recipe book containing all the recipes that are required to build up every single part of you, RNA can be thought of as basically a photocopy of 1 page of that recipe book. So if you were interested in creating a recipe outside of your home in a different kitchen, you might make a photocopy of 1 page of the book and take it with you in order to make that recipe elsewhere. And the same kind of idea exists within our cells. Rather than doing all of our production of our proteins within the nucleus, rather our biology dictates that first a copy of the DNA recipe in the form of RNA is created and exported out of the nucleus into the broader cell. And finally, when it makes its way out to the broader cell, it engages with certain molecular machines called ribosomes that exist within our cell. And these translate the instructions that are on these photocopied recipes into the eventual proteins, which are the, again, elements that make up the structural parts of our body, as well as the little molecular motors, which control everything from muscle movement to neurons firing to your heart beating. And the interesting thing about RNA and its relationship to protein is that the more copies of RNA for a particular recipe exist within a cell, the more copies of a protein can be produced. So you can think about this as if you had a bunch of different chefs who are working at the same time, they would be able to be more productive if each of them had their own copy of the recipe rather than all of them having to take turns with the same sheet that contains the recipe. And essentially what we do in our lab is try to understand what makes up cells, what makes up cell types, makes one cell differ from another cell by studying biology at the level of RNA. So going a little bit more into my particular project, I'm interested in studying osteoarthritis. So I'm sure everyone is at least familiar with the idea of arthritis, but specifically osteoarthritis is characterized by the degeneration of articular cartilage. So basically cartilage that exists at the ends of our bones, at our joints, such as our finger joints, our knee joints, our hip joints, all the basically movable joints in our body. And as this cartilage degenerates, it is no longer able to serve as padding on the edges of our bones at joints. And as a result, the bones that articulate or meet up at that joint are able to rub together. And this leads to low-grade chronic inflammation and quite often pain for people who suffer from osteoarthritis. So obviously, osteoarthritis is a topic of healthy study amongst the genetics field as well as in the biological field in general. And large-scale studies of healthy individuals and individuals with OA have determined that genetics play some type of role in determining who is predisposed to developing this disease. So the basic setup of this type of experiment, which has been done by other people in the past, is you have a large group of people who have osteoarthritis, have been diagnosed with osteoarthritis, and another set of healthy individuals who share characteristics are of a similar age, similar gender breakdown, similar kind of geographical location, and similar ancestry as the people in this group that have osteoarthritis. And then the next step is to basically sequence the DNA to understand what genetic makeup exists within each of these two different groups. So here I've kind of graphically depicted one such result that you might get from an individual DNA molecule from each person in this group. And what you might notice is that while all the healthy individuals seem to have similar structures of DNA within this region, within the osteoarthritis group, there seems to be a variant within the DNA, a certain version of the DNA at this particular location that is shared amongst not all, but a large number of people with osteoarthritis. And by doing this for not just this one location within the genome or the DNA of these individuals, but all throughout every single letter that makes up the genetic code in each of these groups of individuals, they're able to basically identify locations such as this that are associated with osteoarthritis. So locations where if you are an individual who has this particular variant within their DNA, you are also more likely to develop osteoarthritis. And a lot of work is now being done, not only in the osteoarthritis field, but also in terms of trying to understand other diseases. A lot of work has been done to try to explain how these red variants are causing and leading to osteoarthritis or these other traits. So hopefully that gives you an idea that genetics definitely plays a role in terms of whether or not someone develops osteoarthritis. But of course, we all know that genetics is not the end all and be all for whether or not somebody develops this disease. And in fact, environmental factors also play a very crucial role in determining someone's risk for developing OA. So we know this both from observational human studies, knowing that people who engage in occupations or pastimes which lead to increased mechanical stress on their joints, such as in knee joints or hip joints, are more likely to develop osteoarthritis. And we also know this because of animal experimental models within the lab, which make use of either surgical implementations, such as the transection or the cutting of the anterior cruciate ligament or the ACL ligament, or non-invasively administer biomechanical stress to the joints by putting, for example, a large weight on the knee joint. And these all lead to the development of a disease within these animals, most often either rats or pigs, symptoms as well as signs that mimic human osteoarthritis. So it seems like not only does genetics play a role in terms of determining whether or not you have a risk for developing this disease, but of course our environment also plays a very major role as well. So I mentioned earlier that our lab studies RNA, the intermediate molecule between DNA and proteins that are kind of photocopies of the recipes for the molecular machinery as well as structure of our bodies. And this field is called gene regulation, kind of understanding what causes these photocopies to be made and what causes the number of the photocopies that are made within a particular cell within our body. And the genetics field is very invested in kind of understanding all aspects of human biology, both in healthy times as well as in disease times. But I think a kind of subtle, deeper layer that has started to become more and more evident throughout the past few years is the importance of studying gene regulation, not in just any type of cell, but in specific types of cells of interest for a particular trait. So an obvious example of this is if you were a scientist or a biologist who was interested in studying heart disease, you probably wouldn't be wasting your time kind of studying the skin of an individual or the kind of studying gene regulation within a skin cell or a brain cell, but rather you might get a lot more insight into the biology of heart disease by studying the heart itself. But if we kind of extend this analogy further, so beyond kind of this 3D location of which cells you're investigating, and introduce a fourth dimension, that of time, it might also behoove researchers to kind of investigate gene regulation of cells throughout development. So you can imagine Here I've depicted kind of the developmental trajectory of a heart cell, a heart muscle cell. Here on the right is the adult heart muscle cell, and depicted on the left are kind of intermediate developmental stages as a cell divides from a very primitive state into one that is more and more committed to becoming a heart cell. Obviously, if you were able to kind of recruit me or yourself into a scientific study to study heart disease, and a scientist was interested in collecting a sample from your heart, the cells that they would be able to sample would be these late muscle cells, the ones that have already undergone this kind of developmental trajectory and are already committed to their fate. However, it is possible that certain aspects of our biology, such as heart disease, may manifest themselves and may make themselves evident in these intermediate stages. And therefore, it becomes important not only what type of cell you're studying, but also what developmental time point you're studying it at. So for example, it might be important to study a cardiac progenitor cell, a cell that is not quite an adult heart cell yet, but is on the way to becoming one, in order to really fully understand the disease process behind, for example, heart disease. And if we introduce yet another wrinkle to this, the other thing that we also need to consider is environmental perturbations and how they impact the way our cells behave. So an analogy for this would be if you were to study an individual who comes from a part of the world where the air quality is not as good, and compare the expression of certain genes within their lung cells to that of a similar individual, maybe a twin who doesn't live in that part of the world, but lives in a part of the world where the air quality is a lot better, you might see differences in cellular behaviors merely due to the stimulus that these cells are exposed to on a daily basis. So for example, the cell, the lung cell of the individual in a more polluted environment may have a different response or different cellular activity, different kind of programs that are active compared to the one that exists in the lung of someone who breathes a lot more clean air. So the bottom line is that studying disease-relevant cell types, so kind of if you're interested in studying heart disease, studying heart cells, in disease-relevant cell states, so either studying the right point in development or studying the right stimulus, environmental conditions, may allow us to better understand how diseases manifest themselves and how they arise in the first place. So that brings me to basically the basis of what I'll be talking about today, which is this idea that not only do genes matter for this disease of osteoarthritis, but also your environment, the activities you engage in, the amount of stress that your joint cells experience on a daily basis. Both of these contribute and may actually interact with each other to determine whether or not someone develops this disease. So the way you can think about this is if I had an identical twin and I led a fairly active life, but didn't engage in strenuous sports activities, and my twin, on the other hand, became a professional athlete in the NFL, he might be more likely to develop osteoarthritis than myself, even though we share the exact same genetic makeup, even if we both share this risky version of our genetic makeup that predisposes someone to osteoarthritis. So essentially what my project seeks to understand is how does our genetics as well as our environment kind of interact to predispose someone to developing this disease. Of course, ideally, we would be able to, as I mentioned in the previous slide, be able to sample the relevant cell type for our disease. In our case, it would be cartilage cells, since those are the ones that suffer the most degradation in osteoarthritis. However, getting access to human joint tissue and human joint cells is both ethically and logistically challenging. As you might imagine, there aren't a lot of volunteers who are willing to kind of have a doctor or a researcher dig into their joints and take out some material. And therefore, most of the time when human joint tissues are available for study within the lab, they come from individuals who are undergoing joint replacements, either hip replacements or knee replacements. And the discarded tissue is used for research within the lab. However, in a different kind of strategy to still generate these types of relevant cell types within the lab has been becoming more and more popular as a research strategy over the past few years. And it involves the use of what are called induced pluripotent stem cells. So in order to give you an idea of what induced pluripotent stem cells are, I need to talk to you about what pluripotent stem cells are. So if you kind of imagine yourself way back into your history, we all started off as a single cell. We all started off as a fertilized egg within the womb of our mother. And it's amazing to think that that single cell was able to divide itself over and over again, and then give rise to such a diverse range of cell types that we have in our human body today. So we have neurons, which look nothing like that original cell, that original single cell. We have muscle cells, we have cartilage cells, all which now exist and have their own specialized functions, but are completely different from that original cell. And moreover, that original cell had the potential to develop into a neuron. It had the potential to develop into a muscle cell. It had the potential to develop into a chondrocyte or a cartilage cell. And essentially what induced pluripotent stem cells do is kind of rewind time. It takes one of these adult cells, adds a bunch of different factors, and it allows the cell to return back to this earlier state where it had the potential to differentiate or to develop into a different type of cell. So to give you an idea of how this works, essentially adult research subjects will donate their blood. And then from the blood cells, we introduce what are called reprogramming factors to kind of rewind time to turn this adult blood cell, which has committed itself to a blood cell fate, back into that progenitor cell, that cell that has the potential to develop into a wide variety of different fates. And then what we do in the lab is take these induced pluripotent stem cells, the ones that have the potential to become these different fates, and guide them down one of the paths that is of interest to us. So for example, if a researcher was interested in studying heart cells, they could take these IPSCs and differentiate them into heart cells for their study. Similarly, someone who's interested in studying neurons could do the same. And in my case, you are able to differentiate or cause to develop these induced pluripotent stem cells to develop into cartilage cells, which I can make use of in the lab. So a second hurdle that we need to overcome for my project is the fact that in science, we obviously like to be able to measure the amount of, in our experiments, we want to be able to quantify, want to be able to control the types of variables that we're introducing into our experiment. Obviously, if I wanted to introduce mechanical stress to cells, a simple way to do it would be to take a mortar and pestle and then grind up my cells manually. However, that would not guarantee that if I came into the lab on Monday, did an experiment where I grinded up the cells, that on Tuesday, when I did this, replicated or repeated the same experiment, that I would be able to repeat it exactly how I did it on Monday. Fortunately, engineers have developed kind of marvelous devices and machines to kind of allow for the controlled introduction of mechanical strain to cultured cells, such as cartilage cells. The way that these devices work is by making use of what are called flexible bottom cell culture plates. So these are kind of vessels in which we grow our cells in that have a clear, flexible membrane as their bottom. And when paired with this device, which utilizes vacuums in order to stretch that membrane, we're able to introduce stress to the cells, because as the membrane stretches, the cells that are living on top of that membrane also stretch in response as well. And because we're able to control the amount of stretching and the duration of the stretching that these cells experience, it makes it a lot more reproducible. It makes it a lot more controllable as scientists to administer a certain dosage of mechanical strain to the cells that we're studying. So here is an overview of the experiment, which I'm going to be describing the data from in the next few slides. Essentially, what I did here was take IPSCs, or those induced pluripotent stem cells that were originally derived from adult human blood, from 3 different individuals, so 3 different donors, caused them to differentiate down a lineage, which eventually became cartilage cells, and then used the device that I showed on the previous slide to administer a certain dosage of biomechanical strain to those cells. At the same time, I set aside a portion of those cells to not experience the biomechanical strain, to understand what's the difference between a cell that experiences this biomechanical strain versus one that doesn't experience that strain. And in order to study these cells and analyze these cells, I collected gene expression measurements, which essentially just means I collected information about the RNA molecules that were expressed by these cells. And just a reminder, RNA, again, is the intermediate between DNA, the recipe book, and the proteins, which are kind of the machinery and the structural components that make up our bodies. So if I took these cartilage cells from each of my samples and then extracted the RNA molecules, here represented by the little squiggles in different colors, I then measured them using this device called a sequencer, which basically reads the genetic code which is encoded on each of these colored molecules, and then outputs the data that looks like this. So here is a grid of essentially numbers where the columns represent samples. So for example, one sample might come from individual 1, their chondrocytes that were experiencing biomechanical strain, whereas the second column might be the cells from individual 2 that did not experience biomechanical strain. And the rows are genes. So genes you can think about as the different flavors or the different types of RNA molecules. And each of these colors represents the fact that each of these RNA molecules has the recipe, has the information to produce a certain type of protein. And the numbers that kind of populate this grid basically are the number of count-in molecules of a certain type within a particular sample. So for example, in sample 1, let's say this is sample 1, and I've extracted this number of RNA molecules from that sample. The sequencer would count that 1, 2, 3 of these are coming from the yellow gene, and therefore put a 3 next to the yellow gene in the first column. It would also count 1 black, so 1 next to the black, 1 green and 1 blue, 1 next to the green, 1 next to the blue, but 0 orange molecules. So it would be a 0 in that row. And it repeats this for millions and millions of RNA molecules, which are extracted from each of the individual samples, and it repeats it for each individual sample within my experiment. So as you can imagine, that leads to a lot of data. So if you were to think about it in terms of number of dimensions of the data, we see the world in 3 dimensions. The number of dimensions within this data is basically the number of genes, which is on the order of 10,000 different dimensions in the data, which is too much for me to kind of comprehend, too much for anyone to kind of humanly comprehend, although the computer is able to kind of conceptualize that fairly easily. So in order to better understand and conceptualize of this data, fortunately, statisticians have developed methods to kind of take this 10,000 dimension data and reduce it down to a few dimensions, which are human readable, human interpretable, but still capture most of the information that's already located within the data. So what's another way to say this is, if, for example, this black gene had the same exact amount of expression in each of my samples, so all across this row, there were values of 1, 1, 1, 1, 1, 1, 1. There's no difference between each of my samples. Then knowing how much black molecule was picked up by the sequencer for a particular sample tells you nothing about which type of sample it is. It could be from sample 1, it could have been from sample 2. It's not really meaningful for interpreting the data. And what this statistical kind of technique called principal components analysis does is it identifies dimensions within the data that actually explain the most amount of variation. So it ignores all the ones, all the genes, all the colors that don't really allow you to differentiate between individual samples, and instead focuses on those that do. And you can see here that I'm able to reduce that 10,000 dimension data into basically 2 dimensions, which I'm depicting here. The first dimension, which explains 32% of the variance, almost a full third of the amount of variation within the data. And the second, which almost explains a full quarter of the variation within the data. So taken together, they can give you a representation of the data, which explains basically more than 50% of what is actually contained within that humongous grid that would otherwise be impossible to kind of wrap your head around. So the takeaway from this is that here I've plotted each sample kind of as the sample label, what I called it essentially, and colored each of the samples by the individual donor who kind of originally donated the cells that were eventually undergoing stress or not undergoing stress. And what you can immediately see is that the colors seem to go together. All the green ones seem to go together, all the red ones seem to go together, and to some extent, the blue ones also group together in a larger group, but also in a smaller group, there seems to be smaller substructure within this individual. So one big takeaway from this is the fact that the samples, the amount of variation within my data seems to be largely attributed to which person the cells are coming from, which gives you an idea that perhaps the genetics of these individuals plays a really big role in terms of determining what types of RNA molecules, what types of activities that cell is engaging in. And of course, this is like a very qualitative look at the contribution of individual factors to the variation in my data. We as scientists like to quantify that, put a number to it. And here, using a different statistical modeling technique, we determined that the individual as well as treatment status of our samples contribute the most variation into our data. So essentially, if you were to take cells from individual 1 and compare them to cells from individual 2, you would more likely than not be able to distinguish those two just based on the data alone. And you would be right the vast majority of the time. And this is also true for treatment status. So if you were given 2 samples and told that one of them came from a sample that underwent the biomechanical stress, so were stretched, and one of them came from a sample that did not undergo that biomechanical stress and was not exposed to the machine, you would be able to, more often than not, be able to distinguish those two.
Terrific. Thanks, Edwin.
So the next step that we wanted to take was to kind of understand the differences between our 2 conditions. So the cells that underwent the biomechanical strain here on the bottom and the ones that did not. What's different in terms of their cellular behaviors, and can we glean something important, something meaningful from the patterns that we observe? So one scenario you can imagine is that after extracting the RNA molecules from our control cells, the ones that did not undergo strain, and the strain cells, the ones that did undergo strain, you might see that they both yield the same number of yellow molecules. So it seems that even after the administration of the strain, there didn't seem to be a change in the activity on this level, on this one dimension. Well, you can similarly kind of envision a scenario where this is not the case, where there is a difference between the different conditions that we're observing. So in this case, the cells that did not undergo the strain produce 3 green RNA molecules, but the ones that undergo the strain produce a lot more. And this suggests to me, at least, that in response to the mechanical strain, the cell changed its biological system. It started to engage in activities which it did not engage in before, And the way it does that is by turning on the expression of this gene, this recipe, and producing a large number of copies of this recipe to allow it to produce proteins in response to the strain. So this protein could be responding in a way that fortifies the cell in response to the stress so that it's not killed by the stress, or it could be proteins that are designed to move the cell away from its current situation and get out of Dodge to escape its stressful situation. Or you can imagine a bunch of different other cellular activities that could be turned on in response to this mechanical strain. So from my analysis, I was able to identify around 1,000 of these genes, these recipes, that showed differential expression, so differential amounts, between my two treatment conditions. So those are kind of depicted here in the red, with the other genes that did not show a significant difference, were kind of like the yellow situation in the previous slide, are depicted in gray. And the interesting thing and the encouraging thing is that these red genes, the ones that responded to the mechanical strain that we administered to the cells, fall in categories that are related to joint health and joint homeostasis, and also, based on prior studies, have prior evidence for being involved in joint health and osteoarthritis. So to give you an example of that, these are some of the kind of descriptions of the cell, of the genes, of the recipes, of the activities that the cells that underwent the mechanical strain are more likely to engage in compared to their unstressed counterparts. So you can see amongst the list of the cellular activities, response to stimulus, as well as various processes which are involved in organization or production of extracellular matrix, which you can think about as kind of the structure that glues cells together in a cartilage tissue and allows for the development of a strong kind of strong cartilage pad at the end of bones. So in addition to the biological processes that we found were represented by the genes that have found differential expression between our control cells and the ones that were exposed to stress, we also looked to previously published studies looking at osteoarthritis. So in this study, they actually took advantage of some of the discarded tissue that came as a byproduct of joint replacement surgeries and found that different genes were differentially expressed, so had different RNA levels as well as protein levels between high-grade cartilage, so cartilage that showed more degradation, versus low-grade cartilage, or cartilage that maybe has some signs of osteoarthritis, but is nowhere near as degraded as the high-grade cartilage. And what we find is that our differentially expressed genes are more likely than not to be included amongst this group of genes that were also identified by this previous research group as having some involvement in this disease. And we also find this for a different study as well. So I think what this shows is that the system that we've used, which takes advantage of IPSCs or induced pluripotent stem cells, differentiates them into cartilage cells, which are otherwise very hard to obtain from human subjects, and then administers controlled biomechanical strain to them, creates responses within these genes which are important for human joint health, as well as for osteoarthritis. And what I am interested in doing next is taking the small scale experiment, which I have kind of already analyzed, and scaling it up, including more induced pluripotent stem cells from a larger number of donors, in order to, instead of finding locations within the genome, within the DNA, that are associated with whether or not someone is more likely to develop osteoarthritis or not, but rather find locations within the genome that are associated with whether or not a particular cell, a particular cartilage cell, is more likely to produce a large number or a small number of a particular gene, or a larger number of that particular gene. And the idea behind this would be that if the same genetic location is associated with both gene expression level differences and osteoarthritis, one might actually be causing the other. So in this example, we see that this red part of the DNA is more likely to be found in individuals with osteoarthritis, is associated with osteoarthritis, than in individuals who are healthy. At the same time, this same red variant is associated with whether or not a cell within that human is more likely to produce a smaller number of this yellow gene versus a large number of this yellow gene. It could be that this same variant, this same red part of the genome, is causing both of these to happen. It may be causing the cell to produce less of this gene, less of this RNA, less of this protein, and as a byproduct of that, predisposing the individual with that variant to developing osteoarthritis. And that's essentially the next steps in my research project, and trying to understand the relationship between what we're seeing at the RNA level and the relationship that we're seeing at the level of disease status versus healthy status. Right. So I want to acknowledge some people who obviously made this work happen, since I could not have done it all on my own. I want to thank especially members of my lab. I work in the lab of Yoav Gilad, who is an incredible mentor and has been very supportive throughout both my early graduate career and obviously throughout this coronavirus situation, as well as lab members who have provided mentorship as well as support throughout my past 3 years, or two years going on 3 in the lab. I also want to, of course, thank ARCS for their support of my research, my program, and the University of Chicago's Research Computing Center for providing kind of the intensive computational research resources which are required to process the 10,000 dimensional data, which I described earlier in my slides. And I'd be happy to take any questions.
All right, thank you, Anthony, so much. First, virtual and applause on the video. I do think you should leave your screens up because we are likely to have some questions where people would ask you to go back and maybe show some slides again. So let me go into my gallery view mode. I have a few questions that I put in the chat as you were talking, but before I go ahead and do that, does anybody have a question to go first? Oh, Donna, you're muted. Let me unmute you. Oops, Donna, you gotta. Okay, Donna, I unmuted you. There you go.
Well, first of all, I want to say this must be a very attractive field, Anthony, because look at all the young people that you have on your team. There's somebody who can't even walk yet. So this is a really, I think that's pretty impressive. I like that. If you have osteoarthritis and it settles in one part of your body, does that mean that you're very likely to have it in other parts of your body? Or is it something that could find itself more isolated, you know, in your knee, but you'll never get it in your shoulder or whatever? Or if you get it one place, is it likely to hit you everywhere?
Yeah, that's a great question. So obviously this is a topic of study, but there are definitely signs that it's more localized to a particular joint rather than being like distributed throughout your body, such as like rheumatoid arthritis is a classic example of a type of arthritis, which is caused by immune cells attacking your own body. And those individuals are much more likely to develop distributed arthritis throughout different joints in their body.
Interesting.
So yeah. And then in fact, I mentioned earlier that they had genetic studies which associate different parts of the genome with your predisposition for developing osteoarthritis. In fact, if you subset the definition of osteoarthritis to, for example, only hand osteoarthritis or hip osteoarthritis, you see different signals, different parts of the genome which light up, which are associated with development of the disease, which kind of suggests that even on the genetic level, there might be certain parts of the genome that explain a certain type of osteoarthritis, but not another.
Interesting. I asked the question because I have a very dear lifelong friend who has had 2 knees replaced, 2 hips replaced, 2 shoulders replaced. She has hands that make her nuts sometimes, and it's getting harder for her to, move around. But she has said it's osteoarthritis. I have other friends who have rheumatoid arthritis, but for them, for my friends, that's very localized and very centralized in one part of their body, like feet or something. So I think this is intriguing that, it sounds like it might be isolated, but, it's also, I guess, isn't.
Yeah, the interesting thing about osteoarthritis too is, I mentioned back during the definition of osteoarthritis that there is this low-grade chronic inflammation that arises. So inflammation arises within the joints because immune cells are basically sensing that there's some type of distress that's happening at the joint.
Yeah.
And because of the proliferation of immune cells at the joint and the death that's happening of your own cells at the joint, oftentimes the immune cells will start to learn, you could say, that their own cells, these joint cells, are doing damage to the body because they sense the damage. They know that the joint cells are in that area, that there is damage, and they kind of associate the two together. So
Interesting.
it is possible that people with advanced stages of osteoarthritis may develop an immune system that attacks the joints themselves and contribute to kind of like the seeming spread of osteoarthritis from one joint to many joints.
Yeah, interesting. Very interesting. Thank you.
Yeah, I had a few questions. So, like, for instance, the pluripotent stem cells, I've heard of them before, but I didn't realize how incredibly magical this is, because that used to be a huge issue in research, is to get stem cells so you can do these kinds of experiments. So I didn't know that you could actually take somebody's blood and like make that cell go backward in time and make it come into something new. That's just incredible. So I did not know that was really cool. One question I had though was, you mentioned about biomechanical stress versus strain at different points, and so I thought those two had slightly different definitions. So could you elaborate on what's biomechanical stress versus strain?
Yeah. So I guess they are 2 different terms, and then biomechanical stress in general would be any type of mechanical kind of perturbation of a cellular environment that causes stress to the cells. So one type of that is strain, which is a particular stress which involves stretching of the cells. There's also biomechanical compression, which is another form of stress in which the cells are kind of compressed, going back to the mortar and pestle kind of idea. There's also shear stress, which is, if you think about it, kind of the flow of fluids across the surface of a cell, which leads to kind of a stretching of one part of the cellular membrane. So in my situation, I am, as you picked up on, studying biomechanical strain. So the system that we're using stretches the cells in order to introduce the biomechanical strain.
I see. Yes. Okay. Yeah. So yeah, I guess so stress is the overall concept, and strain is a particular kind of stress. So then
Exactly.
you could have, sort of like in airplanes when they talk about shear stress, we're talking about the airflow over the wing or whatever. Yeah, there's different kinds. And then you mentioned, and I guess you did elaborate on this later, but as you were talking first about gene expression, so gene expression means that the cell is making RNA products. So it's actually like outputting RNA, and that's what gene expression is. And so then when you have those little strings coming out of the cell, that's what its outputs are.
So, yeah, so to clarify, the RNA molecules usually do not leave the cell, so do not leave the outside of the cell, but they do enter into the general outside of the nucleus, the central region, into the what is called the cytoplasm of the cell. And what is often exported out of the cell are the protein products of the RNA kind of recipes. I guess what was confusing is that, as you correctly mentioned, I later have a depiction of little little strings coming out of the cell. So this is part of processing the cells. We actually have to break those cells open in order to release their RNA contents. And the reason why we do this is in order to be able to put them through this device called the sequencer in order to measure them, because otherwise they would be inside the cells and inaccessible to the machine.
Oh, okay. And you mentioned that they have this gigantic sequencing computation that you had to do. How long does a sequencer take to run all your 10,000 genes and millions of samples or whatever?
Yeah. So it depends on how large your samples are, or how many, how deeply you want to sequence them. Like, for example, you could think about reading each molecule once, or if you wanted to be extra sure that the machine's not making mistakes, have them double check, read through them multiple times. But it takes on the order of around like a day or two for the samples to be run all the way through. So it takes a decent amount of time for it to do all that.
And if you go ahead in your slides, I've heard of principal components analysis. I studied that a little bit in psychology. So that's a really general statistical technique to kind of sort out what's contributing to variance that you see in a data set. But then you had a cool graph after the PCA, which I had yeah, that thing. Yeah. What is that modeling technique that you refer to there? Yeah.
So this might be getting into the weeds a little bit, but essentially what it does is take your gene expression information, so the values which are within that large grid that I depicted earlier, and then creates a linear model. So an equation for a line. If you remember back to algebra in high school or middle school, y equals mx plus b. There's y, which is the amount of gene expression that you see. X, which is a variable. So it could be individual, it could be treatment, it could be anything. And then b, or m would be the slope, or the effect that an individual has on determining how much RNA molecules you see in a sample. So by kind of creating a model which has all of these components, it's able to estimate the relative effect, the contribution of each of these variables to the amount of RNA molecules you see from the sample. And that essentially is how it's able to determine how much of the variance, how much of the variation in Y is determined by each of these variables.
Okay, so it's basically a linear regression.
Yeah.
Right, okay.
It's a linear model.
And that's an interesting way to visualize it, I guess.
Yeah.
Okay, interesting. Okay, I'll open the Q&A on the floor. Other people have questions? Well, Anthony, I do want to ask you, before I forget, I want to ask you one other thing is, first of all, sort of two kind of questions more about you. When did you get interested in science, from your childhood onwards? And then also tell us how the ARCS award has helped you in your graduate career.
Yeah, certainly. So for the first question about when I first got interested in science, I think I was always interested in school subjects as a kid, but I really fell in love with science, and more specifically biology, during middle school, because I had one teacher in particular who was very passionate about science. I think if she had been a physics teacher and I were in a class, I would be doing physics today. So that definitely ignited this kind of spark of being interested in science, as a way to understand the world around us, to kind of describe things in a way that makes sense to not only me, but also can be used to build models to understand other things that may not be directly related to the topic of study. And that only grew as I moved into kind of middle school. When I went to college, I knew that I wanted to be a biology major, and up till even now, when I'm obviously engrossed in science on a daily basis. Yeah, so I would say that. And then for the second question about how the ARCS kind of scholarship has really supported me throughout this graduate phase of my career, I think it's been tremendous support, not only during normal times, but also I want to say in particular during this kind of COVID madness. So earlier we were talking about kind of psychology of aging, but there's also this, I guess, like theory or model within child developmental psychology, this concept of critical periods, where they last on the order of like around months or so, where it's important for a child to be exposed to language in order to allow for them to develop language skills later on in development. So I like to think of graduate school as also having these analogous critical periods where it's really important for people to be productive, to be engaged in their research, to be kind of working on their projects in order to hopefully graduate in a timely fashion and be successful. And I feel like this whole situation fell at an inconvenient time for me in terms of my career, because it was one of those critical periods where I was just on the way to being very productive in the lab. And then the quarantine happened, so we were told to shut down our lab activities and weren't really told when we would be able to go back to lab. So that was a very stressful time. But definitely having the support from ARCS in terms of like financial support was able to allow me to continue engaging in research even during kind of the lockdown period. So I was doing a lot of analysis of my data during that time. And just knowing that I had this other kind of source of support to fall back on, despite the fact that the university at some point was talking about like a freeze on kind of discretionary spending towards research in order to kind of ameliorate some of the losses that were coming from the effects of COVID. I think it's been tremendously helpful in that regard in terms of allowing me to remain productive in this critical period of my research.
That's fantastic. Thank you.
It is, it's terrific. Terrific. Where, Anthony, can I ask, this is Donna again, where you are in your research, how many more years do you have before you see the light of day beyond a PhD?
Sure. So if all goes well, I should be graduating after 2 years from now, basically 2 years from now. So,
okay. Yeah.
So I'm basically like around midway through my graduate career.
Wow. Is that your PhD and MD?
Oh, just my PhD. Yeah.
Okay.
Then I still have more school after that, but at least I'll have a PhD to show for it.
So you're going to go to medical school after this?
Yes. Yeah.
Oh, wow. Very interesting. Well, now I know who to call when I get osteoarthritis or any kind of arthritis.
So is your plan to, after getting the MD, are you planning to actually become a doctor and see patients, or are you looking more at medical research?
Yeah, so right now I'm planning on focusing the majority of my time on medical research. It's definitely something that when I went into the program, that was the intention. But of course, I would like to make use of my medical training to kind of inform the questions that I ask, kind of inform my research directions. Obviously, as you saw today, I'm already kind of influenced by medical questions in terms of what I like to engage in and with in the lab. So.
Oh, I see. Barbara has put a question in the chat. How will this research apply to osteoarthritis treatment? Mm-hmm. So is there like a gene therapy kind of treatment that you envision, or what? Yeah.
So I guess there are two different avenues which I could see this kind of developing fruit down the line. The first is obviously the gene therapy kind of example. If we're able to identify genes, so going back to the last example I was showing where there was a particular variant or very particular portion of the genome where if you have a certain red version of the gene versus a different color of that version of the gene, you're more likely to develop osteoarthritis. If we're able to identify the genes that are responsible for predisposing someone to osteoarthritis, we could definitely develop gene therapies to kind of correct for that deficit within individuals who have the red version of the gene. The second kind of maybe more preventative kind of aspect of this would be if we're able to identify the interactions that occur between genetics and environment in terms of predisposing someone to osteoarthritis. if we're able to identify people with these red versions of their genes, we can advise them to maybe consider not going into the NFL, maybe consider healthy activities which strengthen their joints and prepare them to kind of weather this genetic predisposition for developing the disease. And hopefully with interventions that come early enough, that we could kind of stave this off and prevent individuals who might be predisposed to developing the disease from actually developing it in the first place. Yeah, so there's both a treatment angle and a prevention angle I can see.
All right, thank you so much. Any other questions from the audience? All right, well then, I think since we're a little bit over time, we'll wrap it up. So once again, let's thank you. I'm going to put my little virtual clapper thing in there, yay. So if any of you can see the reaction button on your Zoom, that's what it's for. Just clap or do a thumbs up to say thank you again, Anthony. It was really enjoyable, fun presentation, really a really good level for our audience here. So it really helps that you did a great job explaining this. So I can tell you're going to be a great doctor and researcher because you're so good at explaining this to a general audience like us. So once again, thank you so much. And ARCS members, we'll be doing another technical talk pretty soon. So thanks for being here at Fun at 5. Thanks, everybody. Bye-bye.
Thank you.
Thanks, Patty.